Good morning, Dr. Sebastian and all the viewer. My name is Kizen Xiang from University Science Malaysia. This video is prepared for the assignment VZT306 Animal Behavior, and the topic is about sexual reproductions and the behavior of weakly electric fish, the family of Mormyridae. Biologists has classified the fish that can generate electric into two groups, which is weakly electric fish and strongly electric fish. The weakly electric fish can generate about 1 voltage of electric through the electric organs. That electric organs can also called as electrocyte. The electrocyte is disc-shaped and located at the structure of the tail. The brain will send the signal through the nervous system to the electrocyte, and the electrocytes will then release the potassium and sodium outside of the body and form an electric field around the body. Electric that generated by the strongly electric fish is high in voltage, and it can use to stun the prey and cause the prey paralyze and consume it alive, or use it as a protection to stun the predator. But in the case of weakly electric fish, the electricity they generate is low in voltage and it cannot use to hunt the prey. Then, what is the functions of generate electricity? In the weakly electric fish, the electric can use as electrolocations and electrocommunications. Electrolocations is to detect the locations and the possible prey and predator surrounding them in order to make the decisions to fight or flight. For the electrocommunications is to communicate with other electric fish around them by interfering the electric field and by generate the different frequency of electricity. The topic is interesting for me because it can help me to understand the role of the electric organ discharge for the reproductions of the weakly electric fish. Other than that, it can also help me to understand the behavior of the different sex of weakly electric fish towards the electric organs discharge for reproductions. Last but not least, it also helped me to understand can the weakly electric fish differentiate the species of another weakly electric fish by detect electric organ discharge. Besides that, the research questions that can be asked in the experiment is that how does the male weakly electric fish electrocommunications to attract the female during the mating? Is there any electrical waveform between the species for species recognitions? And also, is that only the male can discriminate conspecific and heterospecific individual? In the experiments, the hypothesis 1 is that the male can discriminate the conspecific and heterospecific. Besides that, the hypothesis 2 showed that the electric organ discharge mediated the conspecific associate preference during the mating seasons and after the gonadal regulations. Next, we will go to the methodology of the experiment. In the experiment, all the test subjects stay in different aquariums before the experiment is carried out to prevent the weakly electric fish communicate with each other. The species of the test subject is Campylomormis compressirotris and Campylomormis tamadua. Next, the test subject is placed in the aquarium with 3 compartments. Each compartment will have different category of weakly electric fish. In the middle of compartment, the male focal fish will be placed, and on the left compartment, the male stimuli heterospecific fish will be placed, and on the right compartment, the male stimulate conspecific fish will be placed. In between of the compartments, the grid is added to prevent the fish moving around in aquarium. The result of the male focal fish stay in preference zones will be recorded. In the next experiment, the equipment and the methods remain the same, but the category of the test subject was changed. In the left compartment, the female stimulate heterospecific fish is added, and in the right compartment, the female stimulate conspecific fish is added. The result of the focal male fish will be recorded when staying in the preference zones. After previous experiments, the next experiment will be carried out with a different subject. In this experiment, the left compartment is replaced with the dipole carbons electrode with the prey bag of EOD from the heterospecific fish, and the right compartment is replaced with dipole carbons electrode with the prey bag of conspecific fish electric organ discharge waveform. The result of the male focal fish stay in the preference zone will be recorded. In the result of the experiment, the Campylomormyridae compressilosis shows a preference to stay in the zones that near to the conspecific male or female weakly electric fish compared to stay with the heterospecific male or female weakly electric fish. The result of the Campylomormyridae tamandua towards the live male conspecific and heterospecific fish is not stated in the report, but the result of the Campylomormyridae tamandua towards the female conspecific and heterospecific show that the Campylomormyridae tamandua is preferred to stay in the preference zones that near to the conspecific weakly electric fish. The result of the EOD playback with the Campylomormyridae compressilosis it shows more preference toward the conspecific EOD waveform, but in the case of the Campylomormyridae tamandua show no response toward the EOD playback from the dipole carbons electrode. Based on the experiment, the ability to distinguish conspecific and heterospecific is always associated with the match choice. But the match choice is different with the specific recognitions. The species recognitions isolations does not affected by the gender of the species of weakly electric fish. Besides that, the result of the EOD playback stimuli have two different behavioral responses shown by the fishes. The Campylomormyridae compressilosis shows to be attracted to the conspecific EOD playback, but in the Campylomormyridae tamandua shows no response to any of the EOD playback. In the conclusions, both of the hypotheses is accepted, but with a few exceptions. In the hypothesis 1, it is found that the male does discriminate between the conspecific and heterospecific weakly electric fish. Hence, the hypothesis is accepted. Other than that, between both Campylomormyridae compressilosis and Campylomormyridae tamandua, it shows that the EOD wavelength is sufficient for one of them to species recognition behavior isolations, but not another one. Hence, the hypothesis 2 is not fully accepted. In my opinion, for the further or future research, the number of the species can be increased in order to have a more reliable result, and the number of the test subject can be increased as well. That's all from me. Thank you for viewing and stay safe.